In this lecture, we're going to discuss how to do a literature search and how to dissect a journal article. And we'll be using the information we learned last week about research study designs and statistics and how we can apply that information to determine whether a journal article is a good article or not. So why do we need to do a critical research appraisal when we read a journal article? And the reason for this is because just because a study is published and peer-reviewed doesn't mean that it's 100% correct. Studies can get recalled or edited after the fact, after publication. And there's also the problem that approximately half of published medical journal articles have used their statistical methods incorrectly and then made erroneous conclusions based on the misused statistical methods. In fact, this is so prevalent in research that there is a scientist, Dr. Eric Rollins, that has an entire website dedicated to detailing how current research articles have misused statistics and the effect that has on their findings. And this is a fun website to check out if you have some free time to kind of see where science has gone awry and why statistics actually mattered, which is why it was important for you to review your research statistics handout. So one reason why we care about doing this critical research appraisal is that clinicians and policymakers are going to be changing their practice based on the results of published research. So that's part of that evidence-based practice that we want to be utilizing. But we want to make sure that when we're making those policy changes and changing our practice, that we're doing it based on information that's correct. So before we make a change in our practice, we need to appraise the study critically that we're using to make that change. So as you read an article, you should answer questions about the design of the study, the methods they used, and how they analyzed the data and what their conclusions were before you form a decision about whether you need to change your clinical practice. So as you read an article, you want to kind of summarize the study, and you're going to apply the following 4 questions to summarize a research study. It kind of helps break it down into smaller chunks for you because a research article can sometimes feel a little overwhelming to read. So the first question is, where did the authors start? So this is usually covered in the introduction and includes like the main research question. And then what did they do? So this is usually found in the methods section. So what experiments did they do? What was their study design? How did they collect samples? Things like that. And then what did they find from that, from those methods? What were their results? And then finally, what does it mean? So this can be found in the discussion or the conclusions of the journal article. And it basically tells you what the author thought their study meant. And keep in mind that might be incorrect. So you also want to draw your own conclusions based on what you saw of their research, their results as well, and what their methods were. The standard research article has kind of a set format that they're going to have these certain components, and that helps you orient you to where things are in the article. So the first thing is the title and the abstract, which kind of gives you a summary of what the article was about, the introduction, the methods, which can be broken down into subparts such as the subject, the study design, any instrumentation that was used, procedures, data analysis, things like that. The results of those studies, any discussion and conclusions the author drew, and this usually includes future questions they want to pursue, as well as references of where they got their content from in terms of their introduction material and discussion items in their and references in their discussion. And there may be an acknowledgement section where authors can thank grant funding and anyone who was particularly helpful with funding that research. The research title of the article should be formatted to state the purpose or the conclusion of the research. And one of the key things here is that it should draw the reader's attention because this is what they're going to be seeing when they're searching for articles and doing a literature search. So it needs to be clear, and it should grab their attention so they know if they want to save that article to read it later, or if they want to delve a little deeper and read the abstract, determine if it answers the question they're looking for. So the title should be brief, about 10 to 20 substantive words. So in the following example, you can see that we have investigation into the distinct subcellular effects of docosahexaenoic loaded low-density lipoprotein nanoparticles in normal and malignant murine liver cells. And you can see that I was an author on this. But what this basically means is that this particular paper looked at subcellular effects of DHA that was loaded into an LDL nanoparticle and looked to see if that killed normal or malignant mouse liver cells. So someone who's used to reading this jargon would be like, oh yeah, I understand what this is. I'm going to save this. So the abstract is a short, it's about 300-ish words, summary of the article that should include the major aspects of the article. So this includes the overall purpose and research problem, the basic study design, any major findings, and a brief summary of the conclusion. This is like really brief. And what's the purpose of the abstract is that it allows the reader to review kind of the main points of the article before deciding if they want to read the entire study, entire article, because reading an article is time consuming. So if it's not going to provide the information you need, there's no point in reading it. So the abstract kind of gives you a sneak peek as to whether the article is going to be useful for you. It's kind of like the Cliff Notes version of the article. So this is an abstract that does a really good job of showing you the component parts of an abstract. Some journal articles, their guidelines are to have it as kind of one big massive sentence where you kind of have to go and find where these breakpoints are, whereas other journals have the guidelines where they want it to be segmented like this, where we have the background, the methods, results, conclusion, and significance all clearly delineated. But you can see that each one of these sections is only like 2 to 3 sentences long. They're very brief, and that's just to kind of give you a general overview of what this abstract covered. It's not going to be super detailed. It's really just the highlights. The introduction of a journal article should clearly present the conceptual aims of the study. So why are they doing the study and what they hope to get from it? And then a hypothesis should be clearly stated. And I will say that a lot of journal articles do struggle on actually clearly stating the hypothesis. They're not going to just come out and be like, the hypothesis is X, Y, and Z will happen when we change this. What is more usual is that they just kind of go over the aims and what they're hoping to accomplish, and then they go over the results later. Because a lot of times when you do research, you don't know what the results are going to be, so you don't really have a hypothesis of what it is. You're just exploring. And then you're going to summarize the existing literature and include a rationale. So here is the introduction to that paper that we've been looking at, and you can see that this first paragraph is essentially establishing the rationale for the study. In this particular paper, liver cancer is really hard to treat because if you give a patient chemotherapy, their liver's already damaged and you'll give them liver failure. So we need drugs that won't harm the normal liver, but will kill the cancer cells. And then the second paragraph kind of goes over and reviews the previous literature, including the previous studies that that particular lab has done. And then the last paragraph provides the aims of the study. So what they're hoping to accomplish, which it says the goal of the present study is to elucidate the cytotoxic sequelae that occur within cancer cells following LDL DHA nanoparticle treatment. And then it kind of gives a brief little overview of how they're going to be doing that. So this is kind of what you would kind of expect to see from a journal article, but it's honestly pretty rare that they're going to say the hypothesis. So in the methods, they will go over what they're doing the research on. So that could be a cell line, that could be mice, that could be humans, what the selection criteria were, what the sample size was, any potential biases. Potential biases are particularly important when dealing with human subjects. What the study design was it a randomized control trial? Was it a case control trial? Things like that, and how data was collected, any instrumentation and reagents that were used in the testing process, what the procedures were. So for example, if we're doing a drug treatment, what the treatment groups were, how much they were dosed, how often they were dosed, things like that. And the procedure is detailed enough that you kind of know what they did, but it's not so detailed that it's a standard operating procedure that you can follow line by line to repeat what they're doing. So if you look in the example to the right, you can see that we've given the cell lines that's being used. So we tell what the cell lines were, what they were cultured in, what supplements were given, what the incubator atmosphere was, environment was, how we isolated hepatocytes, and then that the cells were plated in 96 well plates. They were treated and then mixed with the reagent, including like the brand and what was used for 4 hours according to the manufacturer's instructions, then read on a microplate reader. And we do have a little bit of statistics included in here that the relative cell viability was expressed as a percentage of the untreated controls. And we could also put in here that significance was determined by ANOVA testing or t-test, things like that. And they'll often have a separate section that goes over what the data analysis was as well. The results of the paper should present all the relevant data as clearly as possible. So the authors are going to include their key figures that best illustrate their conclusions. They might have additional figures that weren't able to fit in the paper due to page limits. That can be included as a digital appendix on the journal's website that can be downloaded by the reader. And you should also state the statistical test used for each figure and table. So the figure on the right used an ANOVA test. It also includes the p-values that were used as part of the to determine statistical significance between the experimental group and the control group. And when you read the figure description, you can actually get additional information about the study design and the methods. So on the previous slide, we showed that this paper did a cell viability assay and it used the different cell types. But in this particular figure description, we can see that we looked at the cytotoxicity of LDL-DHA in 3 different cell types after they were treated for 72 hours with increasing doses, and it tells the concentration span after serum starvation. And the conclusions of this figure, which is that the cancer cells or the TIB75 cells were more sensitive to the drug compared to the normal cells and the primary hepatocytes. And these standard error bars here can tell you the precision of the data. So these can either be standard deviation bars, confidence intervals, standard error, and that will be covered in the statistical analysis section of the paper or in the figure description. And one thing papers can do is have little stars above the graphs that will basically tell you when a group met statistical significance from the control group. This particular figure doesn't show any because it happened pretty early on and there would have just been stars everywhere, which would have been very cluttered to the eye, but it did describe where that happened in the figure description. And one thing you want to do when you're looking at a journal article is you want to look at the figure and the data and make your own conclusions. You don't want to just read the author's conclusions because they might be incorrect. And it also helps your critical thinking skills to try to figure out what you got from this information. So for this particular cell survival, I can see that our cancer cells very quickly died with treatment, whereas our normal cells, it took them a little bit. They had to have a much higher concentration before they got down to 50% cell survival. So that showed that the normal cells were not as sensitive to the drug as the cancer cells, which is what we want because we want the cancer cells to die and not the normal cells. And I can make this conclusion on my own by looking at the figure before reading the author's conclusions. So the discussion portion of the paper should clearly answer the questions that were posed in the introduction or cover the aims, basically what the conclusions of the results were. So the results section really just kind of shows the results and doesn't really delve too much into what those results mean. So the discussion is where you really talk about how the results support the author's conclusions. They might suggest ideas for future research based on what they've determined in this study. And it's a place for the reader to ponder questions like, what were the potential sources of bias? Were these covered by the author in the discussion? A good author should cover those. Can that bias be quantified? Could confounding bias be a problem? And most importantly, are the author's conclusions supported by the results? And this is why you want to look at the results and make your own conclusions to make sure that your conclusions and the author's conclusions are matching, because sometimes the authors will try to overreach their conclusions based on their results, because everyone likes to think that their research study is awesome and that it's the best thing ever. and it's your job as a critical appraiser of the article to determine if their conclusions are supported by their actual results. So now that we've talked about the component parts of an article, now we're going to talk about the most effective order to read an article, and it might not actually be the way that it's presented. So the first thing you want to do as a kind of novice to reading journal articles is to read the abstract. That will give you the overview of what the article will be covering. It also tells you whether that article is going to be useful for you for your literature search. Then you're going to want to read the discussion to try to figure out the conclusions that the author made, and then the introduction to get any background information, the rationale, things of that nature. You could also do the introduction first and then the discussion. Then you're going to look at the results, see if the results match what the discussion was, and look at the methods to determine how they actually got to those results. However, an expert in a particular field, when he's doing a literature search, may read in a different order. So they might read the abstract to determine, is this an article I want to delve into deeper? Then they'll look at the results, make their own conclusions, because they're very used to looking at results. Then they're going to see what the methods are, whether they think that those methods were a good way to approach that problem and that research question, look at the discussion of what the authors of the paper concluded, and they can read the introduction, though honestly they might not even read it because they're experts in the field. They already know all the introductory information. It's not new to them, so they might just skip that entirely. But they're just going to be looking at the results and methods and making their own conclusions and seeing if the discussion matches that. Now it's time for you to start a critical appraisal of an article. So a critical appraisal of an article should include some sort of structured method of how you're going to be appraising the key aspects of the research paper. You should kind of have your formula for how you're reading articles and determining their credibility. And then you also want to do a balanced and objective review of the strengths and weaknesses of the paper. And this will become easier as you become a better consumer of research literature. So some things to consider when you're doing your critical appraisal. So was the study original? What were the study subjects? How many of them were they? How were they selected? What study design was used, because a randomized control trial is better than a retrospective study. Bias, were researchers and participants blinded, because that increases the rigor of it, and what statistical procedures were used, and were those statistical procedures appropriate. So on the following slides, we're going to present some preliminary questions that you can address when you begin to analyze an article. There are approximately 7 questions you can ask while you're critically appraising a research study. The first being, why was the research conducted, and does it add to the literature on the topic? That second question does kind of assume that you have an idea of what the literature on the topic is already, but basically you want to just know, why are they doing it? Is it a good reason? Who was the study about, and who can you apply it to? And was the study of the design sensible and appropriate for the research question they had in mind? Like, did they do a retrospective study when a randomized control trial was really what was needed? Was the data collected appropriately to answer the hypothesis? Did they avoid bias or minimize it as much as possible? And hopefully they would point out where the biases are, because it's a good idea for researchers to have an idea of where some problems may be in their research. And then, were preliminary statistical questions dealt with, and does the data support the conclusions? I think one of the most important things here is, does the data that they showed support the conclusions that they made? So when we're thinking about why was the research conducted and how does it add to the literature on the topic, we want to be looking at, was this study larger than previous studies? Did it continue for longer? Did it have a longer follow-up? Or was it in any way more substantial than previous studies that have been done? And is this methodology any more rigorous than previous studies? So there might have been a previous study that was a cohort study, and now we're doing a randomized control trial, which is considered a more rigorous study design. And will these numerical results of the study add significantly to a meta-analysis of the previous study? So there are certain topics in science that are kind of hotly debated, and a new study can help tip the meta-analysis results to one side or the other. And then another question to ask is the population that was studied different from previous studies? So previous studies might have looked at a predominantly white male population, because that's what's readily available, but this new study looks at a more ethnically diverse population, and that increases its applicability to the world, which makes it a better study than previous studies. So the second question to answer is, who's the study about? So are these results applicable to your practice? So if you're reading a paper that was based in the Middle East and looking at their patient population, it might not be super relevant to your practice here in Utah. Also, how were these subjects recruited? So if you were doing a study on the usefulness of the public library and you're recruiting people outside of the public library, that's going to be a highly biased population because they already had a reason to go to the library. You would be better off going somewhere else to the broader community to determine their opinions on the public library. And then how were people included or excluded in the study? And there should be clearly defined criteria for this. And one thing to keep in mind with these criteria is that it may cause a bias in the study. If you excluded people that should have been included, or you included people that are going to skew the results. So that's just something to keep in mind. And another thing to factor in is, were these subjects studied in real life circumstances? So there are pros and cons to having things in real life circumstances. The pro is that it's more generally applicable to real life because you're doing it in real life circumstances. But the problem is that it has a lot of opportunity for confounding biases that we can't control because the world is messy. And if we do it outside of real life circumstances, more in like a bubble, we create a little research bubble, then it's easier for us to determine that X caused Y. Whereas if we're in the real world, there's other things messing with it and it's harder to make that connection. So there's pros and cons as to when a study should be done in real life circumstances versus a little research bubble. And you also want to make sure that you're aware that your sampling and selection criteria can skew the results of the study. So you want to make sure that when you're reading a paper that you're looking at their sampling and selection criteria and making your own conclusions about whether that could negatively affect the results and the conclusions from those results. So the third question is, was the study of the design sensible and appropriate for the question? So one thing that you may notice as you're reading articles is that the terminology is a little intimidating and confusing. They don't always just straight up tell you this was a randomized controls trial that was double blinded or a cohort study. You have to kind of read between the lines sometimes. So one thing you want to ask yourself when you're looking at a study design is, what are the specific interventions or other maneuvers that were being considered, and what was it being compared with? So this is basically looking at what was the independent variable and what were the controls. And then the next question is, what outcome was measured? That's the dependent variable, and how was it measured? So that's kind of your procedures and methods, and how was it being measured? Does that make sense? Is it going to answer the question they're asking? The fourth question is, was the data collected appropriately to answer the hypothesis? So one thing to keep in mind with data measurements is that subjective data is more prone to error than objective data. So if possible, we want to use objective data because it's less prone to bias. So some examples of subjective data that we can see in research studies are things like symptomatic effects, where a patient is asked to state their level of pain. And that's a highly subjective thing because one person might find the same level of pain to be a 10, the worst pain they've ever felt, and another person might find it to be a 2. There's also functional effects, so like mobility, and that can have some sort of objective measurements, like can they walk 50 yards, how long does it take them, things like that. And then psychological effects, anxiety, and that's their perception of anxiety, or social effects, like inconvenience. And one other idea of social effects is things like quality of life. And one thing that we can do to try to make these subjective effects more quantifiable or objective is to use a rating scale. So you could be like, on a scale of 1 to 10, how bad was your pain? And to make it even more objective, you'd be like, okay, a 2 means I can go about my normal tasks with very minimal inconvenience, and I'm not really stopped by the pain. Whereas a 10 is I'm curled up in a fetal ball and I can concentrate on absolutely nothing else. So that's some way to try to add more objectivity. And then, if possible, you should replace these subjective methods or supplement them with objective measurements. So you could have lab values or mortality data or other things like that, where you can actually put a quantifiable number on something that isn't subjective. The fifth question to ask is, was bias avoided or minimized? So systematic bias is anything that can erroneously influence the conclusions about the groups or distorts comparison. So basically anything that kind of muddies the water in terms of determining what the results of the study was. So whether the study is a randomized control trial, non-randomized, cohort, or case control study, you want your two groups, your test group and your control group, to be as similar as possible, except for the difference that's being examined. So the only difference you really want between those two groups is the independent variable, and then you're going to monitor the dependent variable. And your assessment should be blinded, particularly for the researcher, in order to minimize bias. Because if you're the researcher and you know that a patient was assigned to the treatment group rather than the placebo group, and you get a weird result, you might double check the result. But if they were in the placebo group, you wouldn't. And that can create a performance bias. So you want to be blinded as much as possible to reduce bias. So there are practices to avoid systematic bias, and they vary based on the study design type. So the study design that's the least prone to systematic bias is the randomized control type trial, because you're selecting your group of participants from a particular population, and then you're randomly assigning them to a group. So you don't have a chance to be systematically biased because it's all random. Whereas in a non-randomized controlled trial, you're trying to basically make a common sense analysis to determine if the groups are as similar as possible. So you might try to make sure that the average age is the same. There's the same number of males and females in each group, and just trying to minimize the baseline differences between the groups. And then cohort studies, this is controlled during the data analysis. So you don't have, there's systematic bias in here, but you're trying to control for it during the data analysis with a statistical adjustment for differences. And you'll often see this in papers going like, after adjusting for smoking history and diabetes, the results were this. So they did factor in any differences between the two groups. And a case control study is the most open to systematic bias because, and this happens at the beginning of the study when you're determining what patient is a case versus which patient is not a case. And this happens because you're determining when they become a case, and it's not happening during the determination of the outcome. So there's just a lot more leeway and opportunity for bias to be introduced with case control studies. The sixth question in your critical appraisal is, were the preliminary statistical questions dealt with? So what does that mean? So these answers are usually found in the methods and includes things like sample size, follow-up duration, and follow-up completeness. So what that means, especially for sample size, is that you have enough subjects in your study that you can see a statistical difference between the research hypothesis and the null hypothesis. And this is particularly important when you're doing with human subjects, because you want enough subjects that you can see the difference, but not so many subjects that the IRB or internal review board isn't going to accept the study, because anytime you're dealing with humans, there's the possibility for harm to the humans. So you want to minimize the number of subjects, but you also want to have enough subjects that you get the answer you want. And there are wonderful statisticians that will determine the optimum sample size for a particular study. You also want to look at follow-up duration. So are you going to be looking at this study long enough to see an answer? So if, for example, if we're looking at asbestos exposure and the development of lung cancer, if we took two groups, one that has been exposed to asbestos and one that hasn't, and we followed them up for a year, we're really not going to see much difference between them. But if we followed them up for 15 years, we would see that the asbestos group had a lot more incidences of lung cancer. But 15 years is a really long time, which is where follow-up completeness comes in, where if we let the study go on too long, we lose subjects because they die in accidents, or we lose their contact information, they withdraw from the study, and we can end up losing enough subjects from our study that we don't have the proper sample size to reach statistical significance. So you want to follow them up long enough you see your result, but not so long that you lose them. So it's a delicate balance to determine what the optimum time period is. And the final question of your critical appraisal is, does the data support the conclusions? So you're going to look at the results in the paper and draw your own conclusions. Do your conclusions match the conclusions of the author? And one thing to keep in mind with this is that the author is often an expert in the field, so they might be better able to draw those conclusions, or they might have made a mistake. So a thing to keep in mind is, has the author made any assumptions that they shouldn't have? And that's influencing their conclusions. Now that we've learned how to do a critical appraisal, something that all journal articles undergo is a peer review by experts in the field. And there are reasons why papers are rejected for publication. So an author submits a paper to a journal and they say, hey, I'd like for you to publish my paper. And the journal goes, okay, well, let us get a few reviewers to look at your paper and see what they say. And these are experts in the field. And some common reasons why papers can be rejected is that the study didn't address an important scientific issue or was unoriginal. So they're not going to publish something that wasn't important or has been done 100 times and doesn't really add anything. It's just not worth the time and effort to get it to publication. Another one is that the study didn't test the stated hypothesis, and one way that authors circumvent this is to rewrite the paper with a new hypothesis that the study did cover. And resubmit it. And then a different method should have been used. And if a reviewer comes back and says, hey, we like a good chunk of this paper, but we also think they need to do this test to prove their point, then the authors have a chance to go and do that test and resubmit it with the additional information, or to do a rebuttal of why they don't think that was necessary. Another thing is the sample size was too small, so you just don't have enough statistical power to make your conclusions, or the study was not controlled at the appropriate level. The statistical analysis was incorrect and inappropriate. If that happens, the authors can redo the statistics using the correct and appropriate statistical analyses and redraw their conclusions based on the new results. Or they have unjustified conclusions that were drawn. If that happens, they can rewrite the conclusions. And then there might be a conflict of interest. So this is something that has to be disclosed by the authors when they submit a paper. And this happens when one or more authors could benefit financially from the publication. You'll often see this if a author is a shareholder in a drug company and the paper is about that drug and how that drug is amazing at treating all kinds of cancers. And so if they don't disclose that conflict of interest, then the readers won't know that these results might be biased, because this shareholder, who is also one of the authors, has a financial incentive for this drug to be really amazing because he wants people to buy the drug because then he gets money. So you have to disclose any conflicts of interest. And then there's also just the science was really good, but the paper's just really poorly written. And I have, as an article reviewer in my time, have read some journal articles that were submitted for publication that were just really poorly written, and you couldn't really understand what they were saying, and it just needed to be significantly edited before it was ready for publication. Now that we've talked about how to be a critical appraiser of the literature, we're going to talk about how to do an advanced literature search and why we do them. So one thing you're going to do is perform an advanced literature search, and this helps you find current literature on a topic so you can utilize that information in your professional practice. You can avoid repeating research that's already completed and well established, because there's no sense wasting resources and time on something that's already been figured out. And then one of the key things here is that it helps you formulate and plan your own original research study. So when I was in graduate school, I was trying to figure out whether our drug was causing reactive oxidative stress in lysosomes. And so I looked at other articles that had looked at reactive oxidative stress in lysosomes and figured out how they answered that question. Even though they were using different drugs, the overall methods on how to measure reactive oxidative stress in lysosomes was the same. So I was able to kind of steal their methods and use that as a skeleton framework for my own research using my drug. So it was very helpful to not have to reinvent the wheel. So one key thing here is that a literature search is different than a literature review, which is different than a systematic review. And we're going to talk about these all individually. So a literature search is considered an organized search to find key literature on a topic, and you create a summary of those research findings. But the key point here is that this search and summary is for you. It's for your individual use. You're not going to be publishing it to other people. It's going to help guide your study, and it helps you get information on the topic. So the first step of a literature search is define your search topic. Select your search resources, like what search engines am I going to use, what journal, what article, journals am I going to be using, what search terms am I going to be utilizing on these search engines. And then you're going to compile a search strategy, execute your search, save articles, review those articles, and organize the results of those articles into a cohesive summary. A literature review, on the other hand, goes a step further than a literature search. And it's usually performed by 1 to 2 researchers that are experts in the field or want to become experts in the field, and they qualitatively summarize and analyze search findings on a topic. So they search several databases, find all the articles on that database, read the articles, come to their conclusions, write it up in a paper that is then peer-reviewed and published. So these literature reviews are published into a journal, and they can inform an audience about this particular topic. So they'll select their topic, perform the literature search, develop the argument as after reading all this literature, this is what we think the current status of the field is on this particular topic. Survey the literature, critique it. Here's a key thing, they critique it. And then they write a review of, we looked at all the data, we looked at all the papers, and this is what our conclusions were from what everyone else said. And then they publish that for public consumption. So this is no longer just for you, it's for the world. So a systematic review takes it even a step further than a literature review. So this is a high-level overview of the primary research and identifies, selects, synthesizes, and appraises all the high-quality research evidence on a specific research question, and the goal of this is to guide best practice. So the authors involved in this, they work on this for months to years. They do a systematic search of all the databases. They pull all the articles on that topic. They have clear inclusion and exclusion criteria as to which articles are going to be included and which ones are going to be excluded. And then they critically analyze all the included articles. They eliminate any biased articles, and they may also perform a meta-analysis of the primary data. And the goal of this systematic review is to determine best practice, and their results are then published in a peer-reviewed journal, and they say, after looking at literally everything, this is what we determined to be best practice, or to be the true answer for this particular question. This is kind of a summary between a literature review, a systematic review, and a meta-analysis. So a literature review summarizes a topic that's broad in scope, like cancer treatment. It's qualitative, and it may use sources that are biased, and it doesn't define inclusion and exclusion criteria. It just kind of looks at everything. It's just a review of the literature. A systematic review, on the other hand, is a little bit more stringent. So it usually answers a specific clinical question. So, for example, is vitamin C or chemotherapy a better cancer treatment in patients over the age of 40? And then it defines specific search strategies and how articles will be included or excluded from the review, and it can also include a meta-analysis, but it's not required. A meta-analysis looks at the studies from the systematic review, and then it combines the data from those studies. So they actually have to reach out to the authors of the studies and be like, can you please send me your data? And does a statistical analysis that might overturn the results of smaller clinical trials. So it's looking at all the data from everyone who's ever done this experiment and figuring out what the true answer is. So now we're going to discuss how to perform an advanced literature search. So you want to define a search topic. So for example, if I chose the topic, current and emerging laboratory testing methods for screening, diagnosis, and prognosis in Alzheimer's disease, that's a lot of different topics. I need to identify keywords in the search topic. Number one's going to be Alzheimer's, because I don't care about laboratory testing methods not related to Alzheimer's disease. I want to look at laboratory testings, things that are used for screening, diagnosis, prognosis. Some other key terms would be assay or biomarker. That can help me find articles. So I'm going to find my keywords for that I want to use in my search engines. And then I'm going to go to my search engines and databases. And you're probably familiar with Google Scholar. That one's really common. It's really easy to get to. There's also PubMed, Scopus, Web of Science, Medline, ResearchGate, and also the university library. If you go through the university library, you can log in to your university account, and it will actually give you better access to full articles compared to just searching at home, not through the university. If you're on campus, the other ones work just as great. But if you're not on campus, using the university library is a really great way to access more full text articles. So you can use the VPN or access it on campus or go through the university library. So here's some tips and tricks to keyword searches. You're going to identify your keywords, so things like biomarker, Alzheimer's, assays, prognosis, and then you're going to narrow or broaden your search. So you want to think about synonyms. So assay and test can be the same thing, analyte and biomarker. So you might want to include a search that has a search that uses test, and a search that uses assay to try to get the most results. And then you can also use an exact phrase. So if you put something in quotation marks, then your search engine is going to use that exact phrase and not just the keywords. So it would look for Alzheimer's disease screening in that order. You can also use what's called Boolean logic, where you can use things like and, or not, and they're in all caps. So if I was to put Alzheimer's and biomarker, it would look for both of those terms, and it wouldn't return any results that didn't have both of those terms. Or you can do Alzheimer's, and then you can do test or assay. So then it would look at anything that included Alzheimer's in the terms assay or test. And then you can use the word not. So if you're getting a lot of, if you're doing your original search and you're getting a lot of things that aren't really what you want, and you're like, this is really distracting, I want to exclude this thing, you can say not gene or something. If you're getting a lot of information about the gene, but you actually want like a chemistry biomarker, so you can say not gene, and that would exclude those search terms. So this can be useful to help include more criteria to get more answers, or to exclude things that are distracting. You can also use truncated search terms. So for example, you can use a star. Sometimes the symbol changes based on what search engine you're on, but that star basically ends in fill in the rest of this. I just want these first few characters. So for example, diagnose star, it would then search for diagnosis, diagnostic, diagnostician, and that would give you more search results. You can also use wildcard search terms, where you can put in a question mark. So for example, here in America, we spell behavior with an IOR at the end, whereas a article that was published by an author that is from the United Kingdom, they would have OUR, and that might limit your search terms if you're searching for the American version and they published in the UK version. So if you add that question mark, it gets you more search results. Same for like woman versus women, but the symbol may vary based on the search engine. You can also do what's called an adjacency search, where you can do Alzheimer's and then ADJ3, and the number stands for within how many major words of each other. So if I want Alzheimer's and then I want at least three words later, no more than 3 words later, assay. If I did that ADJ3, that means it's going to look for Alzheimer's and assays close to each other, because they're usually more likely to be related to each other if they're close to each other in the article. You can also do a subject heading search, and this is available in some databases where they have controlled vocabulary terms to describe what an article is about, and this enhances your search by including relevant articles that don't contain your keywords but are still in kind of the same subject. So it can help you find things. So another way to find articles or refine your search is citation searching. So if you find an article that you really like, look at its references and go pull up those articles, because obviously the author of that article that you found useful, it also found those articles useful. So it can be a great way to find primary literature, especially if you're looking at a review article, to figure out some new sources. You can also limit by date range to keep the search to the most recent articles, because if you want to look only what's in the past 5 years, you can limit it to the past 5 years. You also can limit it to English language only. I personally only can read English, so it's not very helpful to me if an article is written in another language that I can't read. So I could limit it by English language only. And then use search limits or filters. So a lot of databases will allow you to limit your searches by publication year, language, and publication type. And you can apply these at the end of the search. You can apply them one at a time to see how they affect the search results. And you can also use search filters that are pre-tested strategies that identify specific literature. So filters include limiting your results. So like, I only want to look at randomized control trials, or I want to look at cohort studies or observational studies. So you can kind of filter if that's available in the database based on what kind of study it was, if you want a more rigorous study to look at. So as you can see, there's a lot of different ways to do searches. And so how do we keep track of it all? Because it can be very difficult to remember where you've searched and what search terms you've used and what the results were. So it's very nice to keep a search activity log to keep track of what your search terms were, where you searched, and what your exclusion or filters were. So once you do your search, you want to save articles that look interesting to a reference manager, and reference managers are great because they can import the PDF, full text version of the article. You can read the article in the reference manager, you can make notes to yourself, and you can search those notes in the reference manager. So it makes it easier to find the right reference when you're actually writing your paper or making a presentation. You can save your searches on databases so you know what you've done. And you can also sign up for emails of new articles that match search criteria so that you're alerted when something new and upcoming has been published. So this is an example of a search activity template where you have your research question. So for my particular research question, it would be Alzheimer's biomarkers for prognosis, screening, and diagnosis. And then I can list what places I'm going to look for articles like Google Scholar, Web Medicine, Scopus, things like that. And then I have my actual log where I'm going to put my where I searched. So like the university library, when I did the search, because if I did it 3 months ago versus today, there are going to be more articles because things got published, and what my search strategy used. So I could put in what key terms I looked for, any filters that I applied, any Booleans, things like that, and then the total number of search results and any comments that might be relevant of like I filtered it by the last 2 years, English, peer-reviewed, full text available, and articles only, because sometimes you get like patents and things like that. So I can keep track of this so I can see what I've already looked for and whether it was fruitful or not. So now we're going to demonstrate how to do a literature search. I'm going to have my research topic on Alzheimer's disease testing. I'm going to identify keywords in there: Alzheimer's, screening, assay, biomarker, diagnosis, prognosis, and I'm going to go to a search engine and see what I see. So I'm going to put in the University of Utah Library website. Go to the Marriott Library. You want to make sure that you're logged in so that you can access full text articles. So we're going to get a new search up here, and then we put in our search criteria. So I'm going to do Alzheimer's, biomarker, prognosis, assay. Okay. And then it has its default settings of books, articles, more, all resources, and I've got 7,000 hits. That's a lot of hits. So I've got some books. Ooh, I've got a novel prognostic biomarker for preclinical Alzheimer's disease. This sounds amazing. I like that one. All right. So this is pulling up everything, and we can see here that we have peer-reviewed journals, we have full-text articles, open access. We can decide if we want what type of resources we want, the locations, years. I don't want 1997, so I am going to set this to 2015, and then I'm also going to set it to English language because I can't read any other language. And I want peer-reviewed journals, and I want access to the full text. I'm going to apply those filters. All right, I'm now at 3,420 articles. Woohoo! All right, so let's see, let's read some synaptic proteins in CSF as a potential novel biomarkers for prognosis in prodromal Alzheimer's disease. That sounds fun. Let's find out what happens if we click it. All right, so on the University Library, it says it's peer-reviewed, it's open access, the full text is available. So this is where we can try Mendeley. So on Chrome, you'll go over here, you click the Mendeley web importer, and if you haven't used it before, it will ask you to sign into your account. I'm going to choose that, and I'm going to add it to my library. So I'm going to add it, and then if we're lucky, it will add the PDF and the citation in one go. If it doesn't, you can follow this link and download the PDF and then add it manually. to Mendeley, but we were lucky. It did download our PDF, so woohoo. And then you could go back and add another one. So let's try this one. There we go. Okay. All right, so I was able to do the PDFs. Now we're going to open Mendeley. And going to sync our library so it pulls it up. All right, so these are the articles we just did. It's downloading the PDF. So sometimes it takes a second. And right now they're showing up in my library, but not in the group. So you can, the green dot indicates that you've read it or not. And then you can drag and drop it into your group. So now it's in your group. And what's nice is that when you're reading it, you double click it to open the PDF. You can highlight. Sometimes on PDFs it gets a little weird about where it highlights. And you can also use note features. So with the notes, I recommend putting detailed notes to yourself about what the content is so you can search for that information later to find the article again in your library. Once you've read the article and made all your notes, you can see a summary of the notes you've written on the side. Once you've finished reading the article, you can go back to your library and then you can mark it as read or not read. And that is how you do a literature search. So you would add in your articles, you would give the abstracts a read, and then do a critical appraisal and determine which ones you want to include in your continuing education project. So go forth and literature search